Okay, so we left off in the last video looking at the ureters, making our way up to the kidneys for how urine is moved. So this is low magnification. Here's that transitional epithelium here. Right, here's where pee or urine would be, and then very thick muscularis that can contract, and so gravity and muscular contraction help move it, and then this connective tissue. All right, the gross anatomy of the kidneys, they are kind of protected. Right, we're looking posterior, obviously. Here's the vertebral column. So these floating ribs come down a little bit, and they then have a little bit of exposure, so they can be damaged from trauma. You have to be very careful with your kidneys. They are only about the size of a large bar of soap, and are paired, of course, as I show you here. So it pretty much controls blood, and so to do this it receives quite a bit of the blood that the heart is pushing, right, cardiac output. And it is retroperitoneal, so it's outside the peritoneum but within the abdominal pelvic cavity. The left kidney is a little bit higher than the right. They're covered by a dense capsule and surrounded by fats. They're kind of really packed in place and anchored to the body cavity with, again, more renal fascia with connective tissue, right? So just really plastered in place when you see them in the fetal pigs, all right? On top of us, of course, our kidneys are the adrenal glands, and you always see these suprarenal, that's the thing, right, superior to the renal. And these, of course, have influence on the kidneys, and specifically in the aldosterone that we'll talk about in this chapter, all right? So when we look at the anatomy inside the kidneys, and you have lots of brilliant models for this in your lab, you've got the outer cortex, which is really this from here to here, here to here, okay? And then you have the medulla here, all definitely emphasized by these pyramid-looking things. And then there is a renal pelvis, which is really a funnel here, all right? There are renal columns, so this is part of the cortex that divides and creates the influence of a pyramid here. So I said the columns provide framework for blood vessels entering the cortex, because that's where you need to get the blood, because the cortex is going to have most of the nephron, and certainly it's going to have the part of the nephron that does filtration of blood. So I got to deliver a lot of blood there. So the columns provide a framework for blood vessels entering the cortex. They create the lobes that are called the pyramids officially. The renal pelvis is an expansion of the ureter inside, and this happens as these major calyces, somewhere in here and here, come together. All right. Renal sinus is really part of the vessels and ureters pass through these sinus. So these are kind of openings, and you'll see they often are packed with fat. And then renal papillae are these, right, the apex of each pyramid is a papilla, or papillae, rather. And that's where, here in the pyramids, all these collecting ducts that we're going to look at in more detail are coming together and finally dumping their fluid into this one tube that we call a minor calyx. All right, so you'll learn all that anatomy in lab and just reinforce, this just kind of reinforces it. So since the kidney's job is to filter, we want to trace the blood flow into the nephron, and the nephron is shown here. And again, there's about 1 million of these per kidney, these nephrons, so somewhere between 1 million and 1.5 million. This is a glomerulus, so a singular capillary bed that I mentioned previously to you guys when I said glomeruli. And so here I say, note that the interlobular vessels are also known as a cortical radiate. All right, so they, in your lab book, you can see these, they sometimes call these by different names. But your renal artery that we left off talking about in blood vessels, each kidney gets their own artery, so there's a left and a right renal artery, branches and becomes a segmental. Right? The segmentals become the interlobars, which run between each medulla. And then when they branch and go along the base of the pyramid, they're called the arcuate, because arcu means Y-shape, and they do look kind of Y-shaped. See that right in there? That's kind of Y-shaped. And then from the arcuate, the next branch is the interlobular or cortical radiate arteries. And from the cortical radiate, shown here, right, you will get the afferent arteriole, so smaller, notice here, that leads into the capillary bed we call the glomeruli. All right, so here again, here's our interlobular or cortical radiate arteriole into a glomeruli. The nephron again is the functional unit, and it starts here and then it moves right to there. So from here to here is a nephron. All right. Its job is to clean blood and balance the constituents of blood. So it's really regulating what's in our blood. And a lot of this returns, as we said, you're going to filter about 200 liters, but you're only going to pee away 2 liters, right? Why is the glomerulus a unique capillary bed? Well, first of all, you can see right here, it's a capillary bed between two arterioles. That's crazy, right? Nowhere else in the body do we see this. Most capillary beds, right, it's arteriole leads to the capillary, and from the capillary is a venule. So we have two arterioles, which means we have two tubes that can easily be controlled. Venules just aren't very good at controlling flow, but arterioles are. Okay, the afferent tends to be larger. It doesn't show you in this picture, but your lab models do show this. Tends to be larger than the efferent, and because of this, right, there is more volume flowing in than it won't be matched in rate. So the size difference in these two tubes affects the rate of flow. So it flows in faster than it can drain because the drainage pipe is narrower. So I say to students, often if you want to really picture the glomerulus, picture it like a sink, though this is all continuous, so it's not quite a sink, but you've got a faucet that delivers fluid and you have a drain that removes it. So if the faucet and the drain are going at the same rate, well then you again are about the same size, then you have this constant rate. But if you've got the water volume turned up and you have more fluid entering than your drain can handle, well, fluids increase in that sink, and that changes pressure. All right. Within the kidneys, the nephron is found mostly in the cortex. As I said, that's where you see these that's why they're called cortical radiates, right? The glomerular capsule is also known as Bowman's capsule, and it's a closed tube. The nephron is totally closed, but it surrounds, and so it actually would close this off too. So there's not this kind of leak. Well, it comes really close. It doesn't close this off, but it's a closed tube, right? So the fluid is going to move from this tube into this tube, and then since it can't move back out, through here, it's going to move forward here. Okay, so and collectively, the glomerular capsule, the first part of the nephron, and the glomerulus are called the renal corpuscle, because corpuscle is a ball, and these look like just a ball when you look at them through the microscope or on your models. So the rest of the nephron is proximal convoluted, and convoluted just means twisted, right? And distal convoluted tubules, all right? And then between these two, and they're named for how close they are to this thing, right? Close, far, okay, so proximal, distal. In between them, they are joined by loops, right? Known as nephronic loops or loops of Henle. And most of the loops are in the cortex, though we have some very long loops that can go quite deep into the medulla or those medullary pyramids. All right. When you look at this under the microscope, right, again, which we ask you guys to do, this nephron is going to receive blood and really the plasma, because the blood cells, remember, they're going to stay in the tube. They're too big to leave. And the nephron is then going to take that plasma and decide, as it moves through these tubes, what things will go back to the blood, right, which we're going to call reabsorption, and what things will go on into the urine, right? And then there's even a process of secretion that we do here in the nephron, and that will be moving something from blood into, right? So if this blood moves on in here, and this blood in what we call the peritubular capillaries, if it still has some molecule or waste chemical in it, we can still move that from the blood into the renal tubule, and we call that secretion. So The filtration occurs here in the corpuscle. The reabsorption is going to be here in what we call collectively the renal tubule, right? And then the secretion is going to occur mostly in the proximal convoluted or the distal convoluted tubules, all right? And said, you know, we also affect blood pressure, as we'll look at that, red blood cell production through erythropoietin and calcium absorption. So these are other things, other functions, but these are the main functions, and they are done by the nephron itself. All right, so this renal corpuscle, so now we're closing, we're really zooming in to see its features and structures. So the corpuscle, which is the capsule, and the capillary bed called the glomerulus, do filtration, right? So here's a low magnification, and here's our capillary glomerulus if we stretch, you know, straighten it out. And sitting on top of it is the wall, one of the walls of the capsule. Remember, the capsule is a cup, so we're looking at this inner wall here. All right, so the inner wall, and we're really magnifying it here, this inner wall is made up of these structures called podocytes, all right? And the podocytes have these pedicels, these little processes, little feet. And so this red showing you here is space where stuff from the blood could flow. So there's these rather large spaces. Now remember, again, blood cells are not going to leave here. They can't make it through this space. Many proteins can't either. All right? But there are a lot of other smaller molecules that clearly can leave here. And if they do move out of here, they're now in the cup itself. All right? So there's a basement membrane, and they show this here. So we're stripping away. In this picture, we've stripped away the podocytes. Here's, you know, again, this is one cell. Big cell that kind of looks like this strange octopus, and here's another cell. All right, so we've stripped away the cell, and there's a basement membrane. This is part of the barrier, right, for when we're moving things out of the blood flowing in this tube through the basement membrane and through these slits. And once they do that, they're in the cup, right, the start or the capsule itself. All right, so I say here again something special, and I want you guys to know this about the blood vessels here, the glomeruli, right, is they are fenestrated. They have holes, and that's what they're representing here. Looks like somebody took a fork and poked holes in it. It's to allow, again, stuff, large, you know, molecules out. So we want to really get as much stuff out of the blood as we can here, all right? And that process of which it goes is called filtration. So we have larger spaces, and I just showed you how we got those larger spaces. And then we have high pressure, and high pressure comes from the fact that this glomerulus is between, right, two arterioles. So we can keep pressure very high in here, much higher than a normal capillary bed. Then I said, note that 10 to 20% of plasma will filter into the cap. So not all the stuff is leaving the plasma, because otherwise then it would become, you know, a solid bunch of cells. That's not going to work. So not everything is going to have the opportunity. So that's why you're going to filter the blood continuously throughout the day. But this 10 to 20% of the plasma that filters into the capsule is going to be, it's going to be changed. And the kidney cells are going to determine what comes back of that. All right, so I say, what else affects this process of filtration? So I said again, the podocytes, the filtration slits, the basement membrane, fenestrations in the glomerulus are all what we collectively call the filtration membrane. So this provides the size selection. So as long as you're small enough to make it through all those holes and spaces, you're going to be in the nephron. I say the movement of stuff from the plasma into the capsule is also affected by charge. Right? Negative items are repelled by these membranes, and positive ones move through. And then there's these mesangial cells that surround the glomerulus, and they help to contract and squeeze a little bit. That affects flow, and they relax a little bit. So they're an added. I'm not, again, going to test you on these concepts here. Just going to read them in your book, and they are something that you need to know about, but not be tested on. I said overall, very important here, filtrate does not contain cells or large proteins, and it is slightly positive in charge.